are probably looking for the link to come in. they take some 5 minutes to connect.
Yeah, no, that's fine. It's end of the day here and Friday. So
you are.
Okay. I'm okay to go beyond 6 o'clock.
Great. Yes, sure. Now we advertise in the website what's the local time of the speaker, for then people get this feeling. So there is your local time. Sure. I'm inviting some people from Australia, so then I think we have to have the seminar very early in the morning here because of the difference in the time.
Yeah.
Okay. So you have a big group there in EBI. And how is the PDB in Europe? I don't know that very well. Don't know if Lucas knows. How many PDB centers are there in Europe? I don't know.
Not in Europe. So the PDB is managed by Worldwide Protein Data Bank, which is a consortium. And the site where I'm sure you go all the time, the RCSB, is one site. And PDB Europe is another site. And we also have EMDB, which is in EBI. So Nelly works in EMDB. And then we have PDBJ, which is in Japan. And we also have BMRB in Wisconsin, Madison, where they work with NMR data. So there are five partners. And actually soon, hopefully, we will also have an associate member in PDB China. And probably PDB India will follow as well. So I think you should also think seriously about if you want to be part of PDB.
Do we need to have a big computer for that, or
you would need? Yeah, you would need infrastructure. You would need. So there are laid out process for becoming first associate member, then becoming a core member.
Okay. We are directed to the New York website, right, Lucas? Rochester, I think, when we put PDB here in South America.
Yeah, I usually use CSB.
Yeah.
Okay.
So I will say a little bit about also in terms of what we are doing as part of what I think should be happening in the future in terms of infrastructure because of because of how
you know 2 weeks ago we had a seminar from Arthur Olson, who wrote Vin Autodoc, and he's developing some models for the cell, a cell model. And I was talking to him that maybe we should have a repository, and we were talking about maybe to host in PDB. What do you think about that?
We already have repositories for both image data as well as some of these techniques that are related to similar to EM, for example, X-ray soft tomography and stuff like that.
Okay.
There is a data resource called Bioimage Archive, which takes both imaging information, but also the soft X-ray tomography or EM.
I understand. But what he is doing, actually, if you go to your seminar in your website, we already posted the YouTube conference of Olson. And what he's doing is a molecular modeling, like if you are seeing a protein in PyMol, but you see the whole cell. You can enter the cell membrane, you can see a virus forming, things like that. And he said that he is working on the format for the file, because then, you know, it's like a PDB file, you produce the image.
Yes.
I thought that there should be a place.
There is. So it's not one place. So for example, the molecular models will go to PDB properly. Probably the image data will, depending on what type of image data or what type of experiment is done, there are archives for that kind of information. So there is Empire and there is Bioimage Archive. There is obviously EMDB and PDB. So at different scales of data, you can go to all these different archives and you'll find data.
Okay. I am already looking at the number of participants because we do like popcorn in the microwave, you know. At some point the popcorn stops popping, and then we can usually take at least 5 minutes. Let's check if we wait a little bit more. Any questions about PDB, Lucas Bleicher? PDB-E? Arthur Gruber, Samir, is connecting from São Paulo.
Hi, Miguelito. Hi, Samir.
Yeah, see, we have people from São Paulo, from many places here. Let me see if I can recognize some more people here. Guys, you could write in the chat, where are you from? Just the city maybe, or the university. If you would like to write in the chat, where are you from? Please say hi, so we can have a sampling of if you are, see, São Paulo, Londrina, this is south of Brazil, Minas Gerais, Rio de Janeiro, Minas Gerais, São Carlos in São Paulo, see, Fiocruz. Yes, Samir, your seminars is a success. You have people watching from many places here, see? Cornélio Procópio. Is someone outside of Brazil attending to the seminar? Recife, Pará, is almost outside of Brazil, north of Brazil.
I just saw Nelly there.
Yeah, see?
I just saw Nelly there.
Somalia?
Oh, Nelly.
Nelly's there. Okay. All right, I think that I can pass the command to Lucas Bleicher. That will be your chair for today. Samir, thanks for coming. It's a pleasure to listen to you. And Lucas, you are the boss now.
Okay, thank you very much. So I'm really happy to have this seminar about structural biology. So as Miguel might have mentioned, we have a postgraduate program which deals with everything from genomics to proteomics and transcriptomics, and but some of us works with structural biology, which is a field of research that has seen some really serious revolution in recent years. First, from the people from electron microscopy, which just had this resolution revolution. So all of a sudden we see those amazing structures from big proteins, big complexes. And now we also seen this amazing new revolution on protein structures done by the computer using artificial intelligence. So I'm really glad that Samir Velankar accepted the invitation. And so we're really excited.
Okay. Thank you very much, Lucas. Can you see my screen? Just make sure you hear me as well. And let's get the right. And I'll keep it like this. I don't know what the process is here, but if you can stop me if you have questions, and we can discuss things as well. So, right. So, so thank you again for the invitation, and I'm going to talk about the new era in structural biology, because as Lucas just explained, the AlphaFold and Rosetta Fold, etc., the AI-based methods for structure prediction. So before I think we go into anything else, I'd like to emphasize why we are studying structures. So they're not just about trying to understand the shapes of these molecules, but it is really about understanding the fundamental biology, these components, their processes, how they work. And what we really want to do is to understand how these molecules and atoms are relevant in a certain biological process. And so we can then address some of the disease-related questions or design new molecules, etc. And so it's really important, and I'll come back to this, that structural biology is about understanding biology and using the structure. So I want to start with a slide on protein data bank, and I think in the next couple of slides it would become clear how this whole AI-based revolution actually is a result of decisions that were taken 50 years ago to start this protein data bank. And so it was launched in 1971 with 7 structures. It's the first open access molecular data.
Aqui, ó, essa aula toda em inglês, ó. Palestrante. Esse cara aqui, ó.
Gente, mas ninguém tá reclamando de que eu pedi luz aqui, não. Será que não caiu?
Ai, meu Deus, caiu.
Aqui, ó. Deixa eu padrão.
Galera, com o microfone aberto, Felipe.
So it's a single archive of all experimentally determined structures, and it's managed by the WWPDB consortium. So that includes RCSB PDB, which many of you may be using every day, PDBJ, PDBE, EMDB, and BMRB. And the most important thing about this database is that from the very beginning, the idea was to store both the models, the coordinates, but also the experimental data. In the early days, the data from X-ray diffraction, and make it available free of charge. So that was always the goal from the very beginning. And as it started, very early on, we saw impact of making this data open access. And the first one I would like to highlight is the paper that came out in 1986 from Chotia and Lesk, where they compared sequences and structures and showed that during evolution, protein structure is much better conserved than protein sequences. And this was really important breakthrough in terms of trying to also do modeling or prediction of protein structure from protein sequence. And I just want to highlight that even before PDB was established, this homology modeling was done in 1969. So this is a first paper where homology model was built by hand. And as more and more structures became available in the PDB, a lot of software was developed to do homology modeling, Modeler, Swiss Model, Rosetta, Fire2. So there are many different servers that allow you to provide a sequence and then give you back a 3D structure. At the same time, around mid-1990s, these 2 papers came out, and these are quite important papers because for the first time, I think they showed that residues that are in contact with each other in 3D structure, they co-evolve. So if one residue changes, the other one compensates by changing. And this kind of is the basis on which all these new developments were done to predict protein structures. And especially because I'm at EMBL, some of this work was done at EMBL in the 1990s, and forms the basis for what we see today. So one aspect of this whole revolution is the data, having open access to data, having standardized data formats, all this which was encapsulated in the form of PDB. The other aspect that was equally important was to have a framework where you can actually assess whether protein structure prediction is becoming better with having all these different groups trying to develop methods. And this is where I think John Moult played a really important role by establishing CASP, and every 2 years having this CASP competition right from 1994, and still it's going on. And this provided the basis for people coming together, trying to assess their methods, and then going back and improving their methods even further. And that's an important aspect of both this open access data and this framework where you can assess whether you are making progress. And this then was helped by having the explosion in genome sequences, the associated protein sequence data in UniProt, and also with structural genomics and many other developments, number of protein structures. So we now have more than 185,000 structures in the PDB. But I just want to highlight the fact that this is still a very small portion of the sequence space that we see in UniProt. So just about 56,000 UniProt accessions are represented in the structures that we have in the PDB. But this was sufficient to make this giant leap in terms of predicting protein structures. And as we all know, in 2020, November, in CASP 14, we saw a really amazing AlphaFold progress in terms of structure prediction. And this is, as John Moult has rightly said, this is the first time a serious scientific problem was solved using artificial intelligence. There were many applications, but this came close to really giving us really good structures. And this was then published in July, and I just want to highlight two important things here. The first one, I think, is that this paper has a supplementary information of 62 pages. And what is amazing here is every possible detail of this method was published in this supplementary method. And at the same time, the code was made available to everyone to see. There was Google Colab established as well. And so just kudos to DeepMind for making not just a amazing method for predicting structures, but also making it available for everyone to use. And as Demis said, this was a big venture for them, and this is kind of DeepMind's most complex piece of AI that they developed until then. So once that amazing progress had happened, I think we had our first meeting on the 30th of March last year between DeepMind and EMBL EBI. And the decision was made to establish the AlphaFold database. There were some really important decisions made in that meeting, and one of them was to follow FAIR principles and also ensure that the data will be open to anyone who wants to use it. So these were quite important decisions made in the meeting we had between DeepMind and EMBL. And then on the 13th of April, we established a team to work on AlphaFold database. We started defining the minimum product definition and also look at infrastructure that we can have using Google Cloud platform. And between April and July, quite a few things were done. For example, an example data file was created manually, and we refined it to make sure that we can get all the necessary information represented in that file. And we decided that we will export data in PDB and MMSIF format. We also made sure that we had the right 3D viewers and additional web components to display information. And the first prototype was made available for testing on 1st of July. And that had search functionality, API, web pages, and with test data that was made available to us. And between 1st July and 22nd July, we refined it, we finalized all the different components, and then the database was launched on 22nd of July. So I just want to highlight the time period. So this was all done in 3 months' time, and this is a process which usually takes much longer than 3 months. And so when we launched the database on 22nd of July, we made sure it's open access under CC BY license. And the first version of the database had about 365,000 models from 21 proteomes, including human proteome. The second version was released in December last year and had structures for every Swiss prot entry. And the reason for selecting Swiss prot entries is because these are proteins which are most studied, and we have really good annotation for those entries. And so that took the number of structures available in AlphaFold database from 365,000 to 800,000. And then another release happened in January, end of January, where the focus was to have structures for proteins from pathogens and neglected diseases. And that kind of covered about 49 total proteomes. So we added about 28 new proteomes, and with about 1 million structures now. And this is, I think, the human genome was one landmark, and this is now, in my opinion, one of the most important data sets since the mapping of human genome. And what we plan is by June this year, we will have more than 100 million structures in this database. So what was amazing, as I started getting some of the test data very early on, I was a little bit skeptical to begin with. And so a couple of things I did was to compare the structures with new structures that were becoming available in the PDB. So these are experimentally determined structures. And this is one example where I wanted to highlight the fact that what I was surprised by was the ligand binding sites. And they were amazingly perfect in a way. So you could see all the side chains perfectly placed so that you could have, for example, this calcium ion or this iron sulfur cluster in this predicted model. And you can also see how good the predicted model compares with the experimental model that became available in the PDB. And this was obviously not part of the training set. Another example that I was surprised to see was this protein where it's a dimer, homodimer, and you can see again the predicted structure somehow has exactly same pose as the dimer as we see in the experimental structures. So somehow AlphaFold seems to reproduce these bound forms of molecules, and that was amazing and surprising to see when we started looking at these structures. So what's the impact of this database generally on life science research? So first and foremost, I think we need to get our head around the fact that we will have structure for every known protein in UniProt database. So that's about 220 million protein sequences. So we will either have it in the AlphaFold database, or we will have a structure which will be very similar in sequence to something in UniProt. And so that's something we have never been able to achieve. As I said, PDB has only about 56,000 UniProt accession, and now we are talking more than 100 million here. But if we look at individual proteomes, again, we can see the impact immediately. So here I have some statistics about the human proteome in AlphaFold database. It covers about 93% of the residues. About 58% of these residues have either very high confidence in the prediction or a really good prediction in terms of the confidence. And about 30 to 40% residues are disordered, and this is expected because we know that about 30% or 35% of human proteome is disordered regions. And comparing that to protein data bank, in PDB, we have about 17% of human proteome residues covered. And so we've jumped from about 17% to at least 58%, where we have really good prediction and almost covered the whole human proteome if we take all the predicted models. But there was another, I think, a side effect of this AlphaFold database and the predicted structures. And this is a quote from this paper, that the unexpected effect of this database is that it visually now demonstrates the prevalence of intrinsic disordered regions. So here we can see very clearly a huge chunk of this protein is disordered. And this is very common. And until now, we were used to looking at PDB structures, which were mostly done using X-ray crystallography, which means you have to have a crystal. And so most of these structures were compact structures, really ordered regions of these proteins. And I think we need to start thinking of proteins as no longer just these compact structures, but this is what is really happening in the cell. We have regions which are completely disordered, and we need to start thinking about those. They have function, and we need to start looking at the whole protein rather than just bits and pieces that we were used to looking at. And this was a very conscious decision when we were discussing about developing the AlphaFold database. The inclusion of the whole thing rather than just the structured part is not just a coincidence. This was a decision made to make sure that we represent the protein as a predicted model, but also give its due to the disordered regions. So what are the limitations of this database? And I think this is really important thing to remember when you are using it. So the first and foremost, I think you have to remember that these predictions are just monomeric protein structures, so individual chains, and we know that proteins are not just individual chains. Many times they are in complex with RNA, DNA, ligands, waters, metal ions, cofactors, etc. And none of this exists in this database. Although, as I showed, many of these may be represented in the models, but but it doesn't exist in the database. This is a prediction of fold and not folding. And I think this is really, really important to remember again. What we have from AlphaFold is a final structure of that particular protein. And we don't have any information or knowledge of how we got there from sequence. So there is no information on folding. It's all information on the final fold. And it's slightly more complicated than just even saying it's a prediction of fold. There is no information on dynamics. And so we have a predicted structure. And obviously proteins are not static. And so we don't know which of these multiple states we might see in this prediction. And even more, in some cases, it might be a mix of more than one state of a particular protein. And so overall, I think although people have done quite a lot of studies on this, it doesn't really, AlphaFold is not validated to predict the effects of point mutations. And it's quite obvious if you go back to what I was saying here. What it is doing is taking a sequence, folding it, giving you the final structure. If you change one residue, it's not trained to understand that one residue might completely make the protein disordered or not fold properly. It will try and get that final folded structure. And so AlphaFold in itself is not a way of understanding or predicting the effect of point mutations. But just 1 minute, let me switch on the light. But this, I think, is going to bring a revolution in biology, because just the scale of structures that we will have available to us. And so many studies of proteins can now start with a 3D model, which wasn't the case before. And you can really use this model as a basis for any new hypothesis. We can use this model to explain maybe previously known data for function, for binding, for specificity, etc. So you can start using these models, but you have to be cautious, and I'll come back to that. And we can design new experiments based on these models. And for example, we can introduce mutations to see the impact, and that can be informed by the model that we see in the AlphaFold database. And so it's bringing in many new opportunities. So first of all, just in terms of bioinformatics, we need to start thinking about how are we going to cope with this scale of structured data. We need to now analyze and compare millions and millions of structures. And that will need new methods, that will need methods that are scalable. So that's something that we need to start thinking about. The other thing, obviously, that everybody is now working on is the protein complexes and prediction of structures for nucleic acid complexes or protein ligand complexes. And I think that's going to progress really quickly. And as I showed, there are many limitations of what we have in terms of the structure prediction with AlphaFold. And so we need to start addressing some of those as well. And AlphaFill is one such way of addressing the limitation that there are no ligands in these models. And so what AlphaFill does is to compare the AlphaFold model with something that is available in the PDB, and then takes the ligands from the PDB. So it's kind of cut and paste job. From PDB, you take ligands and you put them in the AlphaFold structure. But we also need to start thinking about not just having a static predicted model, but how do we understand dynamics? And it's not as simple a problem to solve. And so one of the questions is, how do we even identify biologically relevant conformational states. And that's something that's going to take a lot of work as well. And finally, the real question is, how can we use all these structures to better identify function of protein? Because that's what we really want to study finally, to understand the biology. And this is just one example of the work that was done by David Baker using both Rosetta Fold and AlphaFold 2 in terms of predicting complexes. There is a lot more needs to happen in this space, but this study shows that we at least can start thinking about how will we predict complexes. And this is where I want to come back to structural biology, because when this database came out, one of the things that was questioned was, do we still need structural biology? Do we need to solve structures experimentally? And I just want to say again that the goal of structural biology is about understanding biological function and mechanism of all these complexes. And it's not about just stamp collecting, so having structures and looking at them and saying how nice they look. We really need these structures to understand biology. And so many structural studies can now start with these models. And we can design experiments, as I said, so we could construct and express individual domains. We can design and determine structures of mutants, determine structures of complexes. There are lots of things that we need to do. And as I showed, there are many limitations on these predicted models. So structural biology actually is just entering a new golden age and it's not the end of structural biology. And so, for example, we can use these individual components to study these really large complexes. So this is a nuclear pore complex from Jan Kozinski's lab in Embel, Hamburg. And what they did was to use AlphaFold structures to create some of these complexes. And within months, they could increase the number of components for which they had structure model. And this work previously used to take years to do. And so now we can accelerate this. And you can think about really combining different types of experimental data, sparse experimental data, to actually understand biology using these models. And that could be chemical footprinting, hydrogen deuterium exchange, things like that, combined with something else and these AlphaFold models. And we could answer some of the biological questions using these data. But we can go further, I think, and what we really, I think this is the dream, that we go from just having these individual structures to starting to understand the biological processes at molecular level and at atomic resolution. Won't that be a fantastic way to understand biology? So this is what we call visual proteomics. So you have a cell, you can identify, for example, here they have identified different ribosomes and different states of ribosomes. And just imagine if we could characterize a diseased state of cell and a healthy state and try and understand what's happening at this molecular level. So this is where I think we are going with structural biology. But what I want to highlight now is a real problem in terms of having this scale of data. So as we have seen, many biologists who are not experts in using structures have now access to all these different models. And they have very simple questions like, how do we find structural data? How do we assess the quality of these structure models? Or how do these different structures differ, and is the difference meaningful? And how do I then use these models to actually understand function of the protein, and what are the limitations of both the predicted model or the experimental model. And this is where I think a lot of training needs to be given. A lot of training will be necessary for making sure that these models, both the PDB ones as well as the predicted ones, are used properly to design experiments and understand biology. So in the next few slides, what I want to do is to say a little bit more about accessing structure models, how do we select appropriate structure model, and then how we should be thinking about making structure data more useful. So what kind of tools should we be designing going forward? So even before the AlphaFold DB was established, we were thinking about the fact that we have very small number of structures in the PDB compared to UniProt, and there were all these other data resources like Swiss model, Modbase, etc., which had some predicted structures of high quality. And so we were thinking, how can we bring all this together and make sure that it becomes accessible to a wider user community. And so what we established was this 3D Beacons consortium, where we have Swiss model team, Genome 3D, Modbase, AlphaFold, RosettaFold, and obviously Model Portal. All these teams come together and we defined a common API, common specification to access structure models, both the experimental models from PDB as well as the predicted models, and all the associated annotations. And one of the important parts of this is to provide quality assessment for all the different structure models so that you can look at them and see whether you should be using it for your research. And if you go to 3dbeacons.org and put a UniProt accession, you can then get all the information from all these different data resources in one place. And this is the kind of future of having this distributed way of having data archives, but then making data available through common portals or common mechanisms. But as I keep saying, these are all models. And as with any model, there are some things that are wrong and some things that are useful. And we need to understand what is useful and what's wrong. And this goes for both the PDB as well as the predicted model, and in this case, let's say AlphaFold database. So I want to spend a little bit of time on how do we compare different models. And let's start with experimental structures. And usually when I ask this question, people say they use a couple of things. They use resolution and they use R factor to select a better model. And so here is one example where you have two structures, same protein, it's solved from the same group. The resolution is identical, the R factors are comparable, and so which one is a good structure? If you just look at R factors, you would choose this structure. But if you actually look at the quality of structure, you can see using the WWPDB validation report that actually this structure is better than that one. And so what we need is a way to use this validation information where we have multiple metric to say what is the quality of the structure, what is the quality of the experimental data, etc., when we select structures. And so at PDBE, if you search for a particular protein or whatever keyword you want to search, so here I have example of enzyme hydrolases. So there are about 34,000 structures in the PDB that are hydrolases. So how do we even start thinking about quality of these structures? So what we do is we show the quality of the structure in a simple way. So this first bar shows the model quality, the second bar shows how good the model and data fit with each other. But you can also sorted depending on the quality of the structure to get the best structure. But obviously you want a good structure for a given protein, and not just a list of 34,000 structures that you have to go through one by one. And so if you go to the second tab, which is macromolecules, the results are ordered by individual protein. And then you get the best possible structure for that particular protein. And you don't have to go through all the different structures that are available for a given protein. So here, just one example where this structure is obviously better than that one. And so you would select this entry over that entry if you want to study this particular protein. So this is just a way to help users try and select an appropriate structure for a given protein. So how do we do that in the case of AlphaFold? So apart from predicting structures which are really high quality, AlphaFold also produces two confidence estimates. So one is called PLDDT and the other is called PAE. And in the next couple of slides, I'll explain what those are. So the first one is the predicted local distance difference test, or PLDDT. And what this is a LDDT measure, predicted one, which kind of is a quality measure for the local environment of that particular residue. And so if the structure is correct in the local environment, it will give it really high PLDDT scores. And so how do you use it? So first, before how we use it, it's made available both in the PDB format and the MMSIF format. And anything that is really high PLDDT score, which is above 90, means that not just the main chain, but also the side chains are confidently predicted. Anything between 70 and 90 means the main chain is really high confidence prediction, the side chains not as much. And anything below that is really not a good prediction. Although what has turned out is anything which is below 70 is a good predictor of disorder. So in terms of using the PLDDT score, there are two ways or two things you can do with it. One is to identify a domain or possible ordered and disordered regions. So here, as you can see, these parts are disordered with PLDDT scores below 70 and 50, and these parts is ordered, and the PLDDT scores are above 70. But once you identify this structured part, you can then inquire which part of the domain actually have really high quality of prediction. So as you can see here, the dark blue parts have PLDDT above 90, and which means it's okay to use that, including the side chains, whereas these parts here are slightly lower in terms of confidence. So this is the way maybe to use the PLDDT. So let's look at a couple of examples where you have really high PLDDTs for these domains, and some proteins where the prediction is not as good. But there is another thing here that you immediately see is if you have multiple domains in a protein, how can we assess whether the position or the relative position of these different domains is predicted with high confidence or not? And PLDDT doesn't tell you about the relative position of different residues. It tells you about the local quality of a given residue. So we need a different metric for that, and that's what is this predicted aligned error. So what it is the prediction of the position error at residue X if the predicted and true structures were aligned on residue Y. So if you align structure on residue Y, what's the error in terms of the position of residue X is what is given by this PAE. I just want to make one mention that this is not symmetric metric. So, and we can discuss that later if people have questions. But what it is used for is to assess the relative domain positions. And I give quickly a few examples of how would you do that. So let's look at this particular protein that we just saw and the associated PAE plot. And you can see that there are kind of two squares here. The green is better, the whiter it becomes, the more error in terms of relative position of residues. And so these are residues 0 to 700 on this for this protein on both axes. And so if we look at, let's say, two residues here, 272 and 163, and plot it on the PAE plot, we can see that it's quite green, which means the relative position of these two residues is predicted with high confidence. And so we can say that this is really a nice prediction for this particular domain. But if we look at, let's say, residues which are between these two different domains, so again 272 and residue 429, and plot that on the PAE plot, we see that the error is very high, which means the relative position of these two residues is not predicted with high confidence. And so what that means is these two domains could be anywhere in space. It's just a coincidence and just one solution from prediction that they are placed in that particular position. So what does this mean? Let's take a real example to see how to use this. So this is a protein which is involved in export from nucleus. So you can see it looks like a kind of barrel with a nice hole in the center. And so you could obviously see that there is a possible function where something might pass through that particular the hole in the middle of this barrel. But at the moment you see there is a helix which is plugging that hole, and it's quite tempting to then say maybe there is a function for this, that the helix kind of sits into this hole in the inactive state, and maybe then it is pushed out when the protein becomes active, etc., etc. But let's wait and look at the PLDDT score first. And as you can see, the PLDDT score is very low. So first of all, the prediction that this is a helix itself is not of high confidence. And then let's look at the relative position of this helix. And so the PAE plot, and as you can see, this is the rest of the protein here, and this is that helix at the N-terminus. And obviously the relative position of that helix compared to the rest of the protein is not supported by this PAE plot. So it's just a coincidence that this helix is sitting in that position, and it has no biological meaning. So when you are looking at these structures, it's really important that you look at both the PLDDT and PAE plot. So going forward, this also means that we have to start thinking about how do we make this data available? How do we make these models available? And how do we make sure that these structure models are useful in understanding the biology? And I like this quote a lot. It says, the coordinate themselves, as you can see here, are just points in space, and they may just specify some shape, but they don't tell anything about biology. They don't give you any information on the function unless you actually put some biological context on top of it. Unless you say, this is the protein, this bit here is heme. Now you start understanding what this thing is, and what it might function as, etc., etc. And so you need to enrich these structure models with biological context. And I'm going to talk a little bit about this, and I won't talk a lot about actually the visualization and access. So where are these all annotations available? So let's again get back to PDB to see where all the annotations are available. So PDB data is downloaded and used by many teams. Many different databases use it. And so there is a lot more information and biological context available for these structures in all these different data resources. So what we thought would be a nice effort and a community-driven resource is to integrate the structural annotation. So we already seen the impact of depositing models in the PDB. How about having a data resource where you can deposit annotations, and we can bring all those annotations together, and then the impact will be much higher in terms of how this is used by the community to understand biological function. So we have more than 30 different partners who deposit annotations into the PDB knowledge base, and we combine all that information with the structures and the annotations in the PDB-E and create this knowledge graph using Neo4j database. And we link structures to UniProt residues and the associated functional annotations. So is this actually useful? So if we look at just the database, can we answer any scientific question? And so this is just one example where the question is, are there any small molecules with the same chemical scaffold that interact with the same binding site? And if you then write a query on this knowledge graph, you get an answer where you can see that there are two different antibiotics with slight differences which bind the same binding site. The scaffold, the chemical scaffold is identical in both of them. But you can go further than this and you can say, are these binding sites annotated as catalytic sites? Which residues are annotated? And how conserved are these residues? So you can not just get answer to a really complex problem like this, but you can get even more information in terms of understanding the biology. And so some of the questions we really would like to answer and support our users with are, first of all, what structure data is available for a given protein, which small molecules or macromolecules interact with that protein, and can, for example, structure data shed light on how a particular variant might impact function, and so it's linked to or leads to a disease, or how different structures differ, and what can I understand from that. So what we are trying to do is to bring together these individual pieces to make a sense of what we can understand if we kind of build a nice picture from that. And this is where the knowledge base provides a access to protein structures in a completely different way to the individual PDB sites, where we show individual structure one at a time. So on knowledge base, we bring all that together. So here is one example where you can immediately see how many structures are available for this protein, how many different ligands bind to it, how many different macromolecules interact with it, are there similar proteins available, what are all the relevant publications, etc. So this is all now aggregating all this information in one place. And I'll come back to this superposition after this couple of examples. So what can you understand from bringing together all the different small molecules? So first of all, we map the interaction of all those small molecules onto common UniProt sequence. And so if you just see at this view, you can immediately see there are certain residues which interact with many different types of ligands. So each one of these dots here is different ligand found in many different entries. And so you can start looking at this and understanding where the different residues that are important for ligand binding. You can also look at a expanded view for individual ligands. You can see where the ligand, which residues bind that ligand. And you also have access to download all this information and all the relevant PDB entries with one click. So you don't have to go through and sift for which exact entries have particular ligands, etc. You get that in one click. So let's answer a more difficult question, which is, I have variants, and does it impact function? It's linked to a disease, why it might impact function. So this is one example of superoxide dismutase where it's related to a neurodegenerative disease. You have particular variants that are known and implicated in this disease. And you can start seeing immediately why that might be the case. So these are all important metal ions for function of this particular enzyme. And so you can see that anything you change here is obviously going to have impact on the function. Obviously, not all variants can be explained like this. For example, here, we don't have any annotation that directly will explain the impact, but we are working on trying to understand how we could extract this information from literature. But the way we use structures is actually, we have many structures, and we try and understand how all these different structures are different, and what does that tell us about the function of this protein. So what we have done is to implement a process where we collect all the structures together and cluster them to understand function. And this is one example where it works beautifully. So we cluster all the different chains that we have, and here you find two different clusters. So first of all, we have collapsed the problem of having to look at 20 different chains versus just having to look at 2 chains in this case, for example. And what we find is those clusters are actually biologically meaningful. So one of those clusters is a ligand unbound state, and the other one is a ligand bound state. And so immediately you can start deriving some biological knowledge looking at these clustering. The other thing you can do in terms of ligands is you can look at all the different ligands and where they bind onto the protein. And so here I'm showing you an example of fragment screening experiment for SARS-CoV-2 3C protease. And immediately you see where there is a binding site, and you can see that on the sequence as well as on the structure, and understand how various different fragments bind in this binding site. So what we are trying to do at PDBE is to rethink the way in which we should make this structure data accessible. And there are a couple of components to it. The first component is to actually aggregate all the structure information, and that's done using this 3D Beacons network, where we collect all the structure models. Through PDB knowledge base, we collect all the annotations, and then we bring all this together in this knowledge graph. And what we are trying to do is to create different views of this knowledge graph. So I showed you the view based on individual proteins. We are working similarly on creating view for individual ligands or complexes. And the idea is to help users actually derive biological knowledge from all the structure data that is available. So in conclusion, I think I want to say that the open access to macromolecular structures and sequence data was key to all these recent advances. And any field that wants to use AI, the first thing to think about is actually data sets, and having data sets that are well annotated. The second thing is abundance of this structured data is really going to bring in new era, both in structural biology, as I showed, as well as biology at both the molecular and cellular level. And advance in structure prediction is going to really bring in data-driven revolution in biology, medicine, and transform basic and translational research and life sciences as a whole. And with that, I want to thank the PDBE team, the team from DeepMind, and take any questions. Thank you very much.
Thank you so much for this presentation, Samir. It's really interesting new things to look at. I was just making a comment with a colleague that we have to redo a lot of our classes in structural bioinformatics with all this new information, new analysis. And well, I have some questions myself, but first I'd like to open to the students and others who might. So if anyone can't use audio, you can type the question in the chat. We can forward them.
Yeah, just raise your hands or open your camera. We can wait for questions.
So there is a question about whether these structures can be predicted and deposited to PDB. The answer is no. PDB is about experimental structures, and we are not going to take any predicted models. But you can deposit. So there are two things here. So if you are just creating a couple of models and publishing a paper, you can deposit them to Model Portal. And if you are doing it on a large scale for a proteome or a couple of proteomes, then I think the best way to do it is to have a local beacon, 3D beacon, to integrate with this whole network of 3D beacons that we have established. And we do have a client package that will allow you to quickly spin up a 3D beacon and then integrate with the rest of the beacons that are already present. So those are the two ways, depending on what actually the scale of your project.
Do you pretend to have a server for predicting a protein? I would suggest that people could put on demand a given UniProt sequence. So then you would only run the server for UniProt entries that people would need for some organism, a butterfly, whatever. But it would need to be a complete UniProt sequence. Then you could probably attend this person and do the modeling.
So we might end up in a situation where we will have structure for most UniProt sequences anyway in AlphaFold database. But you can also use all these Google collabs that are really excellent if you want to do a couple of predictions. Yeah. Or you can take the AlphaFold and install it locally and do it as well.
Sure.
One thing that I was curious, is there any case in which the prediction went completely wrong? And if in that case, if you understand why it happened?
Yes, there are cases where it can be wrong. And I didn't show you slides, but it also depends on, for example, if there are enough sequences in the multiple sequence alignment to support prediction. So that's one obvious reason why the prediction might go wrong. And we remove all those from the AlphaFold database. So we don't have cases where the MSA wasn't enough of enough depth. And so there might be a problem. But there may be other reasons as well in terms of not having enough examples in the PDB for a certain type of packing. Although I have to say that there might be new folds here, which means that AlphaFold database or AlphaFold 2 hasn't just learned about what's in the PDB, but it can actually predict new packings as well. But the depth of MSA is a reason for not having a good model.
Oh, speaking of this new interaction, one thing that I think you commented in the slide was that at the moment it's not capable of finding post-translation modifications and things like that. But I was just talking with Richard Garrett, who's in the audience, about this new interaction which has been shown recently, in which you can have bridges between cysteines and lysines. And so people found out there are actually many of those structures which already had them in the PDB, but it wasn't much talked about. Now they're being discussed. So their work on this.
Absolutely right. There are no post-translational modifications, although in some cases you would see the side chains in a place where you can put in the post-translational modification. And AlphaFill is one place where, for example, they are also going to put in the glycosylation as part of the AlphaFold structures. That work was done by John Aguirre in York, and he's working in collaboration with Robbie Houston and Tassos Perakis and making this AlphaFill database available. But it's also interesting, the paper I showed in my slides is analysis of all the intrinsic disordered regions, and they have also mapped a lot of post-translational modifications, which actually are many of them are in the disordered regions as well. So there are few
some people with the hands on.
Yeah, Antonio Mauro.
Yes.
Antonio Mauro, go for it.
Hello, Dr. Samir. Thank you for the wonderful talk. I just was wondering if you could talk a little bit more about the amount of unstructured proteins that you mentioned. Do you believe it's something that there is like a gap still in AlphaFold, or do you really believe we have that big set of unstructured protein in nature? I just like to hear a little bit more. And thank you.
So based on the studies we have done on the human proteome and the predictions we have, the unstructured part in the AlphaFold predictions and the prediction of IDRs, they match. So we know that 30 to 40% of human proteome was expected to be disordered. And if you take the predicted structures, that's the kind of number of residues or percentage of residues that we find disordered. We've done, I think there is a lot more analysis people have done, and that paper that I mentioned, I can put the DOI in the chat, is a really good analysis of human proteome and disordered regions in human proteome. So it looks like AlphaFold predictions actually are also predicting the disordered regions.
Can I contribute to that, Dr. Samir? See, not every human protein exists since ever. Many proteins arise when we were a fish. So if you pay attention to that, mostly all the proteins that we share with bacteria, they are fully globular and completely predicted. The coverage between UniProt and PDB is 100%. But when you go further and further in the evolution, you start having proteins that have a structured suprasecondary core and those regions. So it's if you related the existence of these regions to the period of origin of the protein, you are going to see that you have a great correlation.
Yeah, that's a really good point. And as I said, I think the nice thing of after doing this is there is a lot more analysis that needs to happen and validation that needs to happen of all these structures that we have in AlphaFold database. And also a lot more analysis of the IDRs. But it's a good thing that IDRs are now front and center in terms of all these predictions.
Lucas? Bruno now?
Yes, Bruno.
Yes. Hello. Hello, Dr. Samir. Thank you for a great presentation. And I have a question about the AlphaFold quality of prediction. Do you think in the future, maybe we can merge methods of AlphaFold combining with the knowledge modeling or common ab initio methods, or do you think it will surrogate the other methods and we will use this instead of other methods?
So I think that the great thing that has happened is because the code is now available, a lot of people have taken that code and are playing with it, and that's how things develop. So if you remember the within days of the AlphaFold paper becoming available and code becoming available, immediately people tried out doing complexes and they changed certain things. And there is now a effort called OpenFold as well, which is going to tinker even more with AlphaFold, and all this is good. I think the more we develop it, the better. So I don't think this is going to stop someone from developing something. I think this can form one of the ways in which we proceed. Obviously, there are other ways which we need to predict complexes. This is single proteins. AlphaFold multimer is not as good as AlphaFold. And so obviously there is something missing there. And so, yeah, everybody should be looking at this and trying out different things.
But you are providing computational power. If we use another method, you have to pay for the GPU's processing. Yeah. You are doing that for free.
Yeah, obviously. So I also say why have AlphaFold database? Obviously, apart from the scientific reasons, it's also climate reason. We have all the structures calculated once, and so you can use them. You don't need to again go and calculate them and spend power and use GPUs. So it's also for green reasons. It's good to have this database.
That's great. We go João Paulo, no? Or Bruno? I don't know. Lucas, you call.
Yeah, João Paulo.
João Paulo. We are going left to right because it's easy for us. João Paulo, go for it.
Thank you for your exciting presentation, Dr. Samir. We from Viral Metagenomics, we are quite excited by the possibility of using AlphaFold to uncover what we call the dark matter of the metagenomics. But the problem is sometimes we have a symbol sequence that has a potential viral origin with an entire ORF, but it does not have any similarity with nothing known in the databases. So this might be a problem for the prediction, right?
Yeah.
Do you think that using AlphaFold can help us to infer something about the function of these potential viral ORFs, or it would be still too complicated for this application? Thank you.
So it really depends on what you get. And this is why I explained how you might interpret what you get as well. So I think if you get a structure, you need to look at whether the quality of that structure merits any further use of that structure. And also what you derive in terms of, or what you infer from that structure needs to be validated experimentally anyway. And I think that's the important part. So this is where I go back to all this talk about, oh, whether we need more experimental data and things like that. Yes, we do, because these are still just predictions. And I think we need validation, both in terms of structures, but also functional validation. And I think that's where actually we can make a huge progress now, because people can do different types of experiments which provide functional data and validate these models with that functional information. And I think that's the exciting part of this.
But he can give a try of running locally, right? To see if it gives some good results.
Yes. Yes.
Right, João Paulo?
Yeah.
Okay. I think it's the turn for the president of our association of bioinformatics, Marcelo.
Hi, Miguel. Thank you very much for you and Lucas to arrange all these seminars here. And Dr. Samir, thank you very much for your time to come to present this for us. And I'm just gonna make some stop by here, because the next one to talk is my dearest professor, Arnie. Arnie was my first supervisor in academic world. It's a few years ago, and it's through my heart to see you here, Arnie. Yes. But my question is more directed, and it's the same direction that Miguel has done before, and it's about having like an API to condense or to gather information from the server. So we have talked on this, and the other part is using the AlphaFold. Do you have some missing entries from UniRef? Are you planning to put this? Because every time we want to use UniRef, our sequences are on this missing sequences. It's more than Murphy laws.
So the next update is related to UniRef 90.
No.
So we will have more than 100 million structures, because we'll have reference sequence from UniRef 90 clusters. So your sequence may still be missing, but you will have a model which has 90% sequence identity.
Okay. We're trying to using this dodge. Yeah. But thank you very much for your time. We talk later. Thank you, Miguel. Thank you, Lucas. Cheers.
Thank you.
Dr. Arnie.
Oh, boa tarde, Miguel. Okay.
Boa tarde.
I have a different sort of question. I mean, your talk was very informative, and we got a lot of information from it, really fundamental stuff. But when this incredible progress, this AlphaFold came out, I mean, it scared all the structural biologists, first thing. You know? All the crystallographers were thinking, okay, we're out of jobs. Yeah. That was the first thing. And we did something, the reverse sort of engineering. What we did is number of structures which you couldn't solve. We generated models using AlphaFold and used to solve the structures.
Yes.
Which worked quite well, you know, and there were some errors and all, but you could refine it in a few cycles. And rather than spend 2 years trying to solve the structure, you spend 2 days.
Yeah.
So it's much faster. And my question is basically, does this mean that crystallography will be outclassed now?
I don't think so. As I said, I think we're entering a new era in structural biology. What that means is, I think every method will be used, but you will have to first think about the biological problem that you want to solve and use the appropriate method for solving the biological problem. So for example, both EM and X-ray now are used for fragment screening. That's not going to change. There are many things that these models don't have. And so I don't think any of these methods are going to stop. It's just how we employ them is going to change.
It's just that the modeling is going to be much faster now. So maybe we'll have to work faster.
And so you need to maybe we will get answers to our biological questions much quicker.
I mean, without doubt, it's going to change biochemistry and medicine dramatically because we'll understand pathways, we'll understand everything, these things.
Yeah, I just want to show a slide based on your comment, which I didn't include in my presentation. So, I think these were kind of the first reactions. And as it says, this is, you go through these 5 stages of grief, and now everybody has settled down and understood that these are just models, and this is just another tool that we now have to actually do progress in terms of understanding biology.
Yes, it might just be models, but it answers a biological question.
Yes. Yeah.
But you know what, Samir? If I were doing a bench work, I would look to the proteins that AlphaFold did not solve completely, and I would drive experiments to solve those proteins, with a cryo microscopy today, right?
Yeah. Depends on the question. And this is where I really wanted to emphasize training, because what has happened is, because we had very few structures, it was okay for people to use them because they were all experimental structures. The problem now is you are going to see more than 100 million structures. And that means most biochemists, biologists, geneticists will have a structure to look at. But we are not training people to look at structures in a way that they can use them properly. And what needs to change now is, at undergraduate level, we really need to start training them on using structures, which has not happened for a long, long time. And we have very, very restricted kind of curriculum on structural biology. But that needs to change, because otherwise everybody is going to look at these structures, derive some hypothesis based on that, and it may be completely wrong because they don't know how to look at these structures and use them.
Sure. But I think that people now should look at the AlphaFold database to impose demand on, let's do this cryo microscopy, let's solve this structure because it's not good there, right?
Yeah.
Maybe you could rank the proteins for people to do bench work to solve them.
Yeah. And also just do bench work to understand more function based on these predicted models.
No, function too, sure.
Yeah.
Function too.
Yeah. So lot of things can be done. Really exciting times.
Lucas, you have questions?
Yeah, I just saw this this comment on the chat section about training people here. You can be sure that's going to happen. By the way, next semester we have this course on structural bioinformatics, and I was just talking to Rafaela that we both are just updating all our material to include.
So Lucas, we are organizing a webinar on trying to find out how what's the impact of this, how we need to change the training material, what is missing, things like that. So I think you should join that webinar and provide feedback, because one of the plans is to then produce material to help with this type of changes.
So is that already available?
No. We are going to, we are planning it. So the discussion is going to happen, and then we will start a project to actually develop material based on the feedback we get.
Oh, nice.
We here are under Cabana, right? So anything that you organize in Cabana reaches us.
Yeah. So Kath is part of that. Kath's team. So we will. You will know.
Sure. I have a big question here. that demands computation, but I don't know if it makes any sense. I mean, with those so many GPUs available after you are done with the full UniProt, of course, do you think that's reasonable to run a short simulation, molecular dynamics, to collect how the protein looks like after some relaxation? What do you think about that?
You have to be careful. I don't know how these disordered parts will behave in a simulation. So if you are just going to take really structured proteins, like we have in the PDB, then obviously you would get similar kind of information.
Sure, I would start with the crystals, with the solved structures. But my question is, does it make any sense? Because now you have GPUs, so you can do that. Would it be nice to have the structure after molecular dynamics together with the initial crystal? Maybe. It's a question for the future, of course.
So I have always been asked, so every time people start thinking about the different states and dynamics and things like that, and my answer always is that you can always have many structures which look different. The real question is not that. The real question is which of those are actually meaningful. And that's a real problem. If we have many structures, does that mean they are all meaningful? Even experimental structures. So even EM grade, by definition, gives many states, right? But does that mean all of them are meaningful? I don't know the answer. And I think it's really difficult to answer that question. So we can always do simulations. I think the question is not whether you can do simulation. The question is, how will you use it, and whether it's going to lead to something meaningful, and how will you then know that it is meaningful?
And how to choose one or a few to publish it.
Exactly.
Maybe a short pilot with some postdocs trying, you know. Because maybe in the future that could be something useful for the
so one of the projects we are now working on is actually trying to understand how do we cluster and identify different states of complexes, individual proteins, etc. And how do we then understand the biological meaning of those different states? So how do we use all this annotation that we have in knowledge base and cluster these proteins, and can we then link these to really understand biology? So that's going to be an exciting project, and a very difficult one at the same time.
Okay.
Richard.
More questions? Anderson, Richard. Alphabetically? Anderson, then Richard, okay? Anderson, you have posted a question here? You are talking, but you are muted, Anderson. Spacebar. No?
Sorry. So I posted the question, but in a certain way, I think that the professor already answered, but I will ask again, just for sure. What do you think about people that's still creating models with other software than AlphaFold? Are they losing time or not?
Well, again, I would go back to why they are creating those models, because AlphaFold is creating models for certain things, but if you have expertise in a certain field, you may want to use that to get a model to explain something with experimental validation. And so I don't see why we should abandon all the other methods. I think every method will have its own particular advantages, and so depending on what you want to do, you want to use the appropriate method. And that goes for prediction as well.
Richard Garrett, welcome.
Hi, Miguel. Thank you very much. Hi, Samir. That was a beautiful summary of where we currently stand in the structural biology field. I was interested a bit in how we got to where we are now. And what is your opinion about the importance of CASP as a catalyst for getting to where we are now? Do you think that AlphaFold was developed more quickly as a consequence of the very existence of CASP? Because it's a fairly rare initiative to have a field in which there is this objective analysis of the state of play every 2 years. I'm interested to know, would AlphaFold, in your opinion, have been developed less quickly if CASP had not existed?
AlphaFold wouldn't have been developed, in my opinion, because if you talk to Demis or John Jumper, the two things they say were critical for them to choose a problem to work on was one, data set, which is PDB, and second, a framework that allows them to know whether they are making any progress, and that's CASP.
So is there do you think there's a lesson there for other fields?
Yes, and this is what, so this is why I showed this slide, that without this and this, there is no AlphaFold, and so if any other field actually wants to get into this AI and make use of it, the two things they need is, first of all, data sets, well curated, standardized data sets. And also we have to think about how different or variation in the data set as well. But those are kind of going into much deeper then. But data sets is one thing. And the second thing, a framework that allows you to evaluate progress. So those are two critical things.
Thank you.
You know, I'm a user of the second best yet, the CI tester now. That better AlphaFold in the other, the one CASP before, right? The server from Yang Zhang was almost there, like 60%, but the idea was to try to do that, right? I still compare the AlphaFold with the CI tester prediction. So I think that this competition was really great.
Yeah, yeah. And if you actually want to look at Cameo, which is a server maintained by the Swiss model team, where they weekly, so this is again another important aspect in validating all these structures as well. So they continuously look at each week's PDB release and see what new structures have come out, compare them to all the different predictions, and that includes AlphaFold database structures, but also Rosetta Fold and many others. And that's one way for us to know whether the AlphaFold predictions are good in terms of the experimental structures as well. And I think we need to continue to do that for some time, because in terms of actual validation of AlphaFold 2, before, let's say, last December, it was only the CASP targets, so a very small subset of structures. And now we are getting many more structures where, for example, you can start with AlphaFold model to solve a structure, things like that. But we need to continue this for some time to build that confidence that actually AlphaFold structures are good structures in different contexts as well. And so I think that's one of the things we need to do as experimental structural biologists.
Okay. Lucas. You know that we reached 120 people because there are some people that are watching in the YouTube link. So we got a big audience today.
Any further questions?
Time to thank you, Samir, for this wonderful afternoon here. Almost night in there, right? Thanks a lot. It was a pleasure to listen. It was very important. We taped, so many professors will use the recording for their classes. So it's really nice. So thanks a lot.
Thank you very much. Thank you for the invitation. I enjoyed it a lot. Thank you.
Yeah. Lucas Blasher.
Okay, so thank you very much. As we said, we have a lot of work to do around here. So lots of new stuff to tell the students. So someone was asking where the recorded video can be found. I think we can share the YouTube link before we close this.
It's going to now, yeah.
So here it is. So for those who want to rewatch it later. And so, well, thank you very much, Tony. Well, I'm excited about hearing about this new material you are preparing.
Yeah,
so,
thank you.
Thank you very much.
So please join that webinar and give us feedback on what material you're producing.
You know what? If there was no pandemic, we are all going to Carnival here in Brazil. Tonight is Carnival night already here.
So
we hope someday we can invite you to come here, to come over, Samir.
Yes, very much looking forward to it. That's one continent I haven't yet visited.
Yeah, we'll invite you soon.
Yes.
Right,
Thank
Marcelo.
So
thanks a lot.
Thank you.
Thank you, guys. See you next. Good day, good night, good evening.
Bye.
Thank you.
Bye, people.
Então, Miguel. Pessoal, só queria lembrar uma coisa aqui, que nós estamos tendo eleição da B3C. Tem os sócios que ainda não entraram lá e votaram, entra lá no site, na área restrita do site, que vai até hoje no final do dia. Certo? Isso aí, bom carnaval. Miguel, bom carnaval. Não caiu muito no samba.
Eu vou me fantasiar de Zorro aqui. Eu tenho uma fantasia de Zorro e uma de Vedete. Eu estou para escolher qual das duas eu vou usar.
Vou me fantasiar de pijama assistindo televisão à noite em casa.
Tá certo.
Arne, que delícia ver você, Arne. Quanto tempo, cara?
Vixe, eu estou pensando, quando foi, que ano foi isso?
Não pensa não, Arne, que a gente fica triste. Pensa não.
Eu vejo o Arne todo dia aqui nas bancas.
O Arne que me colocou nessa vida acadêmica e falou assim, estuda que é legal fazer pesquisa, tá vendo? Culpa dele.
Você não tá bravo comigo, né?
Não, acho que não, tá tudo bem. Até que valeu pelo início.
Obrigado.
Isso aí, gente.
É isso
Paula
aí,
Kuzer
galera.
ali, Paula Kuzer ali. Oi, Paula.
A Paula tá aí?
A Paula tá aí, tô vendo aqui. Não sei se ela já saiu também, né, pessoal? Beleza. É isso aí, Lucas. Agora nós vamos, que nem falam os Teletubbies. Como é que falam os Teletubbies mesmo?
É hora de dizer tchau.
É hora de
Isso.
dizer tchau. Vai lá, galera. Um
Tchau,
abraço
gente.
a todos. Um abração pra você.
Abraço.
Tchau, galera.
Valeu,
Tchau, tchau.
votem lá. Carol e Sávio. Tchau.
Tchau, tchau.